This is part 3 of Chapter 6 lecture, and I wanted to backtrack just a minute to talk a little bit more about ribosomes. So an important thing about ribosomes that you should know is that the DNA, or the genes coding for RNA, the ribosomal RNA, have the greatest commonality among all living things. And what that means is that there is very little difference in the DNA sequences of the ribosomal RNA that you find in humans versus what you would find in bacteria. So what that tells us is that this function, the building of a ribosome by a cell, is so important to life that every cell does it almost exactly the same way. It's something that has been conserved through all of the evolutionary adaptations through time, and it's something that is shared by all living things. Another important thing about ribosomal RNA and the ribosome itself is that if you just change one base in the DNA code for ribosomal RNA, it will cause the RNA to not work. The ribosome will not work with just one mutation. So again, the importance of ribosomes is really illustrated by those facts. So now going back to the endoplasmic reticulum, where we left off. So the function of the endoplasmic reticulum is actually, there are a few different ones, and there are two types of endoplasmic reticulum, and each type has a different primary function. So the rough endoplasmic reticulum's main function is to process proteins, and the smooth endoplasmic reticulum manufactures membranes. So we'll look at that a little more specifically, because I don't want you to mix up the two. And so we'll look at which one does which in just a minute. But the structure of the ER is that it's a membrane. It's like a long matrix or a webbing of membrane with an inner area within the membrane called the lumen. And that membrane is connected to the nuclear membrane, or what we call the nuclear envelope, and it actually extends throughout the cell. It's a very large structure. And so because it's connected to the nucleus and the nuclear membrane, and because it's part of this protein synthesis system that we have in the cell, it's part of the endomembrane system. These are pictures showing the two different types of endoplasmic reticulum. You can see the rough here on the left in this picture, and then the smooth on the right, and also how they're connected. And there's a small like channel connecting the two. And this is another picture showing rough in this one you can actually see the ribosomes studding the outside of the membrane on the rough endoplasmic reticulum, and then the smooth would not have those ribosomes. This is an actual photograph showing that same thing. So in this case, it's reversed. The rough is over here, and you can, again, if you look closely or zoom in on this with your computers, you can see the little dots on the outside. Those are all the ribosomes on the outside of this rough ER. And you can see over here the smooth. So now the rough endoplasmic reticulum, its main job is to not just produce, because really the ribosomes are producing the proteins, but to modify the proteins for whatever their function is going to be, either on the cell membrane or out of the cell. So they are going to, where the little yellow penguin is saying here, which cells are going to have a lot of rough ER, what you're going to find is that any cells that are doing a lot of protein secreting are going to need a lot of rough endoplasmic reticulum. And the way that they're going to do that is by packaging the proteins in transport vesicles so that they can be excreted or sent out of the cell. So a type of cell in the human body that would have a lot of rough ER would be a pancreatic cell. because the pancreas, their main job is to produce digestive enzymes that are secreted into the digestive tract for obviously digestion. And so a pancreatic cell is in the business of making a lot of proteins. And those proteins are specifically enzymes used in digestion. So you're going to find in that type of cell an awful lot of endoplasmic reticulum, and particularly rough endoplasmic reticulum. Another thing that you should know about the rough ER has to do back when we were talking and learning about, or you were constructing your cell membranes. So you started to see how the cell membrane has proteins, and particularly glycoproteins, that are embedded in it. And those are really important as we get into Chapter 11. Those are very important in cell-to-cell communication in the functioning of a cell and signaling molecules. So where that is occurring and what is happening is the protein, once it's made, is actually made on the side through the rough ER. The ribosomal that's bound to the ER will actually produce that protein and put it inside the lumen of the endoplasmic reticulum. And while it's in there, a carbohydrate will be added to the protein. And that process is called glycosylation. An example of this, a common one that is made, is an oligosaccharide gets attached. Oligo just means few, and saccharide means sugar. And so an oligosaccharide is often attached to a growing polypeptide chain. And a branched oligosaccharide is composed of a combination of 3 sugars, just glucose, mannose, and acetylglucosamine, which can be and is modified in the rough ER. So this is done first. This is attached in the rough ER, then it's put into a vesicle. So you can see that occurring right here. And then that vesicle is sent over to the Golgi. And then the Golgi will do some more folding of the protein and stabilizing it so that it's ready to be then sent to the cell membrane for its job. Now, the smooth endoplasmic reticulum has other jobs. This is where lipids are going to be made, and particularly phospholipids are made. So the smooth endoplasmic reticulum is really considered the membrane factory. So it's where the phospholipids and the portions of the cell membrane are actually being constructed. And how that happens is actually the endoplasmic reticulum, remember, is just simply a membrane surrounding a space called the lumen. And that endoplasmic reticulum membrane can actually bud off and transport to other parts of the cell that need new membranes or need a repair. So that's how that is working. Another job that the smooth ER has that's very important is detoxification of cells. So, for example, alcohol detoxification, the breaking down of alcohol so that it doesn't kill the cell, occurs within the smooth ER. So, for example, you would not be surprised then to learn that a cell that has a lot of smooth ER would be, for example, a liver cell. So a cell that's a liver is involved heavily in filtering the blood and detoxifying the blood. And so those types of cells are going to need a lot of smooth ER. A couple other things that the smooth ER is responsible for is metabolizing cholesterol, also for storing calcium ions, and also to hydrolyze glycogen. So if you remember again, hydrolysis is the breaking apart of molecules, and glycogen is the storage carbohydrate form of glucose. And so before a mitochondria can actually break down the glucose that is coming from a glycogen store, the glycogen needs to be hydrolyzed within the smooth ER. So again, this will occur a lot in the liver. Okay, so once these proteins are modified in whatever way, and not just proteins, it's also these other things like that we're talking about, the breaking down of different molecules or the detoxification of different things. Sometimes those building blocks will need to be sent out of the cell. So other processing will occur in the Golgi apparatus. So the ER buds off whatever it's been making into a vesicle and sends its product over to the Golgi apparatus. You also hear this called the Golgi body or just simply the Golgi. I think of it as a shipping and receiving center, and I always think of UPS. So it's where UPS Central is. It finishes whatever modifications need to occur. It can modify the products of the endoplasmic reticulum. It also manufactures different macromolecules, and then it sorts them and then packages them into more vesicles. So the Golgi apparatus is also made up of membrane, and so it can bud off vesicles in order to transport what it has made or modified out of the cell or to a different organelle within the cell. So the types of cells that you're going to find a lot of Golgi apparatus in would be any cells that specialize in secretion. So if you're looking at a cell, this is probably not the best model because most usually you will find the Golgi very close to the cell membrane. So it will be closer up here because its job is to send things out of the cell. And so it actually is going to be, and when we say send things out, we mean secretion. So cells that specialize in secretion, you're going to find those in the endocrine glands. Any cells that produce hormones are going to need a lot of Golgi apparatus. Things like the pituitary, also the pancreas, the adrenal glands, the testes, the ovaries, any of the exocrine glands, mammary glands, sweat glands, anything that's doing secretion will have a lot of Golgi apparatus and need that. This is a picture and a photograph actually showing a Golgi apparatus. And another thing that is important to know about the Golgi is it has sides to it. It has a cis face and a trans face. And so the trans, if you think of transport, is the one that faces the membrane. It's the one that is sending the product out. So transporting it out. The cis face is what receives the whatever molecule, protein, whatever it is that is being processed essentially. And so the transport vesicles, oh goodness, I wish it wouldn't do that. The transport vesicles from the ER are going to come into the cis face of the Golgi, and then the ones that are leaving the Golgi are going to leave the trans face. And the caverns that you find within the Golgi are called cisternae. This is just showing how that budding off process works. It's exocytosis. It's in this case, budding off with the proteins encapsulated within the new vesicle, and then that carries them over to the Golgi apparatus where then endocytosis will occur. So this vesicle is surrounded by membrane that can join with this membrane and fuse with it, opening up it to allow the contents to flow in. And the reason this is important is because, for example, perhaps these proteins need a specific pH that's not being found in the cytosol of the cell. Or maybe these proteins are very hydrophobic and the cell cytoplasm itself is very water filled. It's very